Greeting my colleagues. Allow me to express my gratitude for the organizing committee to providing me with the opportunity to present at this excellent symposium and to introduce to you some of the results of our research. The challenge of developing of original synthetic pharmaceutical product is inherently multidisciplinary in nature. Its resolution begins with the design of the molecule structure of the future active ingredient of the drug. It is only from this point that chemical initiate the development of a rational synthesis method and analytical super. In the subsequent stage, biologists join the effort, conducting tests on the biology activity in vitro and vivo experience within the framework of preclinical research. The pivotal and highly responsible stage involves the clinical trials with the determining the potential for future progress towards the development of industrial technology for the active ingredient and the pharmaceutical product derived from it. Realization of this scheme demands a substantial amount of time, approximately 10 years, and significant financial resource, around $10 billion. Personally, I represent there the Scientific School for St. Petersburg State Technological Institute. This is oldest technical university in Russia. As Dmitry Mendeleev, a professor of our institute, said, if without science, there cannot be modern industry. When without it, then cannot be modern science. I have the honor belonging the famous scientific school founded at the Technological Institute by Professor Semyon Vukolov, a favorite student of Professor Dmitry Mendeleev. This main problem, facting of our teacher and us until 1991 for the research and development of technology for the production of components of energy-saturated substance and materials. This is nitro and nitramino derivatives of the carbocyclics, heterocyclic, aliphatic series, capable the participant in the processing on combustion and detonation. For me, studies of high nitrogen heterocycles such as TETRAZOLE, first discovered at Uppsala in 1885 by Vladimir, have always been relevant and interesting. Subsequently, some energy-saturated tetrazole derivatives were obtained and used, which have not lost this relevance today. As my teacher, Professor Lev Bagal, said, did not get a good explosive? Check it out. It is possible that you synthesized a good medicine. This instruction allowed us, after the collapse of USSR, to traverse our scientific foundation and qualified personnel of science and technologies. The slides show some samples of medicine that we have developed and presented to pharmaceutical market. We are currently making effort to implement our scientific and production effort in the interest of import substitution of medicine. This problem is very relevant for Russia today. The purpose of this study, computer prediction and in vitro study of antiviral activity of heterocycle systems containing thioperanachenylene and tetrazole moiety. At this stage, in accordance with the terms of reference, we are looking for drug candidates that is effective against the pandemic dangerous influenza A virus, nor the swine flu. Promising target in this case may be various elements on the virus life cycle, from the absorption of the virion of the surface of the membrane of the attacked cell to the phenocytes, to the formation and release of new viral particles. That is why today we have antiviral agents, the active ingredients of which have a diverse molecular structure. In the present study, we pay attention to the phenolene derivatives which have found application to the drug therapy of various dangerous diseases of human and warm-blooded animals. According to new publication of our Indian colleagues, relatively inaccessible thiopyranochenolene derivatives are very promising object in medicine chemistry. This is thiopyranochenolene. As you now understand well, it seemed interesting to us to obtain and study thiopyranochenolene derivatives containing tetrazole rings in the molecule. Our choice was influenced by the fact that the number of modern medicine for various purposes containing tetrazole fragment in the molecules to active ingredients to increasing every year. These are the active ingredients of antibiotic, antiviral, antihistamine molecules, as well as representatives of a new generation of antihypertensive drug, losartan analogs. Every year, in Food and Drug Administration published a list of active ingredients of new drugs recommended to development by the pharmaceutical industry. At last, one formula of the list contains a tetrazole moiety, just like the quinoline moiety. Such as a variety of diverse biological activity in sperm is explained by various factors. For example, the ability of tetrazole to act to isosteric analog of cis-amide and carboxyl group, which is an important condition for the metabolic stability. Another important factor is the ability of the nitrogen atom on the tetrazole ring to form multi-center hydrogen bonds, which contribute to the formation of strong enzyme substrate complexes. The target's compounds were obtained in a two-step synthesis. The first step, synthesis of azimuthine 1 and 2, which contain tetrazole rings by action regioisomeric enalkyl 5 amino tetrazoles on 2 mercaptophenolene 3 carbaldehyde. It is a good results from two these organic chemists on this slide, Andrei Khramchikhin and Maria Skrilnikova. Thank you. The second one, synthesis of thiopyranochenolene 3 and 4 containing regioisomeric enalkyl 5 amino tetrazyl moiety by the Mikhail Asa-Morita Baylis-Hillman tandem reaction using azimuthine 1 and 2 and phenylpropanol. The structure and identity of all compounds were provided using a complex by modern instrumental methods, including X-ray diffraction analysis. The study of biological activity of compounds 3 and 4 included a combination of computer methods and in vitro testing. Analysis uses PAS online 24 of compounds 3A and compounds 4C showed low probabilities for the compounds to exhibit antiviral activity. However, Vladimir Vasilievich Poroykov told us that low values of the probability factor PA can indicate the novelty the object being studied. Research was continued using protein ligand complex of compounds 3A, 4A, and rimantadine with M2 channel. Computer modeling using the Schrödinger suite software package. Docking analysis showed that compounds 3A and 4A directly block the M2 channel of the viral protein. This is work of Maxim Gureev. On this site, we can see ligand interaction diagrams for compounds 3A, 4A, 4C, and rimantadine with M2 channel. Here we can see that the less potent compounds 4A compared to 3A has increased strain energy with indicated a low affinity due to unstable binding, also estimated by glycemic score value. Compounds 4C demonstrated high activity due to more stable hydrophobic contact with the M2 channel. The propyl substituent is oriented towards the lysine residue laid to 6 of chain B and C, as well as ALA 3O, providing a more favorable binding within the M2 channel's cavity. The cytotoxic and antiviral properties of the synthesized compounds were tested in vivo against influenza A, Puerto Rico virus. The component inhibited a high selectivity index than rimantadine. This work used a group of Dr. Zarubayev in Pasteur Institute. Conclusion and recommendation. The first, in accordance with the computer prediction, the synthesized thiopiranochinolin with tetrazole moiety exhibit moderate in vitro activity against the influenza A Puerto Rico H34 virus, MDSK cells, dog kindly cells. Research in this direction confirmed the promise and should be continued in the direction computer prediction, synthesis, and testing of the antiviral activity of few analogs of previously obtained compounds containing larger substituents on the nitrogen atom on the regular isomeric tetrazole ring. For example, normal butyl, isopropyl, tretbutyl, etc. There are the co-authors of this presentation, as well as the organization they represent. Their specialization, along with the scientific profile of the organization, confirm the multidisciplinary nature of such research. And thank you for attention.
Yes, thank you. We have one question, or already two questions. Dear Vladimir Aronovich, do you think the influenza A virus will develop resistance to your compounds as it did with remantadine?
This is a standard etalon in this the similar investigation, because we see that our compounds is some more good antiviral activity comparing the remantadine.
Mm-hmm. Yeah, and another question, dear Vladimir.
Yes, it is problem in laboratory area, but it is not problem in the world, because remantadine use it and continue use it as one of the first antiviral compounds among us. Our Latvian colleagues in Greenfix contact with all forage contact and pay and pay this to America, France, Anglia, and other one.
Yes, and two other questions are related to biological target. How did you identify the biological target, and are you considering any other protein targets for antiviral drug development against influenza A virus?
This virus H1N1 is one of the serious problem for antiviral therapy all medical people in area. And this is standard we have in Pasteur Institute, not we, you understand that it's, I pass over to Dr. Zarubayev, is one of the great specialists in antiviral control of our country and forage. This is standard etalon for testing the new compounds and to be contact with him a long of time. And now we continue this investigation on our compounds, and some our compounds go to next step. The first my slide, you are maybe understand me, and we have some compounds in the finished step of development of new medicine. Yes, we have some problem in GMP final stage, but we have a good experience in resolution this, and I think we can, including our research in this and our production experience for a solution the problem importer substitution of antiviral medicine in our country. We will solution together to specialists in molecular design, to specialists in investigation of activity in laboratory scale, and we have save some production possibility in this area. You see some generic and original compounds with we present to pharmaceutical